All right, it's 2, so let's get started for today. And we're going to pick up. So there were some questions left on Wednesday's PowerPoint, but since I put up the answers for those anyways, I'll let you try and do those on your own time. And if you have questions about why the answer was the way they were, uh, please contact me. We can discuss it at that time. But otherwise, we're going to pick up where we left off on Wednesday and talking about lipids and move on to other compounds that you would find in the membrane. And we start with steroids. So what a steroid is it's a molecule found in eukaryotes, and it's made out of the cyclopentane, let's see if I can pronounce this, cyclopentanyl parahydrophen, move that out of the way, phenanthrene. Yeah, that's a mouthful. Basically 4 rings. So all steroids have that basic structure. And then based on the modifications that happens to these rings, they have different properties. So the most well-known steroid is cholesterol, as shown here. Has a little bitty head group and then these long polar tails. So 30 to 40% of the plasma membrane is made out of cholesterol. And what these cholesterols do is that when they're in the membrane, they first go through a little bit of a change to form a cholesterol ester, but their purpose is to modulate the fluidity of the membrane. The way I kind of think about them is like they're like wood planks. So if this is your membrane, those are supposed to be lipids. Cholesterol is almost like a big slab. And the more cholesterol you have, it will push against the membrane and make it more solid. So cholesterol, as you increase the concentration, the fluidity of the membrane goes down, just becomes more solid-like. So it's a way for eukaryotic cells to actively modulate how fluid their membrane is at any given time. Plants, even though they are eukaryotes, they don't really have that much cholesterol, which is good because when in our diet, if we eat too much cholesterol, we will have heart problems. So it's good that they don't have cholesterol, but they do have other steroids that do the same type of thing, in that they're made out of this cyclopentane, yada yada yada, and they also help to modulate their cell membrane as well. So that's our steroids, all made out of that basic structure. And then we have a question here. How are steroids and lipids in the plasma membrane similar, and how are they different? So I'll just give you like a minute or so. Think about that, try to answer, and then I will give the answer here in a minute. All right, so hopefully you were able to come up with the answer, as this is kind of basic biology at this point. But to be in the plasma membrane, your molecule needs to be made out of, have two different parts. Needs a polar head group, nonpolar tail. We call these molecules amphipathic. Probably misspelled that, but amphipathic. In that you have a polar and a nonpolar part. So cholesterol kind of has that, where they have this little head group. It's not that polar, but it's still polar enough to be in the membrane. If you're just talking about sterols in general, they don't. But the sterol that's going to be in your membrane the most is cholesterol, and does. And they have a big nonpolar part as well. So that's how they're really similar. But other than that, there's a lot of differences. One, cholesterol has the fuse rings. Lipids have the carbon tails, like we talked about. The carbon tails are a lot more flexible and can have double rings in them. The rings are really rigid, and their chemistry doesn't change much. Cholesterol is going to be a cholesterol, going to be a cholesterol. You're not going to modify that. While lipids, you have a lot of different classes of lipids. So more varied, more static. But same overall kind of amphipathic behavior is the big similarity there. And they also have some different functions, right? Like I said, cholesterol is modulating the fluidity. Lipids themselves can do that, but that's not their main goal, because you actually have to take out lipids and put in new lipids to modify that. With cholesterol, it's taking out cholesterol and adding cholesterol, but at least it's the same molecule at that point. So that was our little refresher on lipids and cholesterol. And if you have a question, feel free to stop me at any time. And I can go and address your question. But otherwise, let's continue on. So outside of cholesterol and being in the plasma membrane, steroids are usually hormones. And so they're usually released by different organs. And what they'll do is they'll travel through your bloodstream, bind on receptors on the cells, and lead to large changes in the cells. And these steroids are all made from cholesterol, which you can kind of tell because remember, cholesterol has this tiny OH group, and your general steroid is just the 4 rings. So all of the hormones that we're looking here all have either an oxygen or rather an alcohol or this C double bond O group. If we look at to see what some of these steroids do, we have your glucocorticoids, cortisol being the most popular one. And these can do a lot of things. So they affect metabolism, inflammatory responses. It's what you release when you're stressed out. Aldosterone or other mineral corticoids, can't talk right now. They help to excrete salt and water that's in your kidneys. So when you have an excess of that building up, you release this hormone, which starts the whole cascade, starts the whole process inside the cell. Then you have your sex hormones, testosterone and estrogen, sexual development and function. And all of these are going to be in your adrenal gland, where they're going to be created from the cholesterol and then released throughout the body. And they all have different functions. There's also vitamins. So it's just not cholesterol, just not hormones, but also vitamins are all derived from steroids. And if you look at this vitamin on the bottom left, you can kind of tell it used to be a steroid, or is derived from a steroid rather. It's just missing the ring here. So the ring got opened up, but it's still steroid based. The precursor to it was a steroid or cholesterol. So these vitamins, vitamin D being the most popular example, are all from steroids. So vitamin D2. Now let's look at these vitamins, right? So here is vitamin D, and you see there's an R group here. This R group can be two different things. It can be 7-dehydrocortisol, which is shown right here, or ergosterol, which is, I got those mixed up. Ergosterol is right here, 7-dehydrocholesterol is right here, right? And for vitamin D to really be effective inside your body, you need to go outside. Yes, I know. But sometimes you do in this horrible Texas weather. And well, right now it's great, but in the summer. And then you need to get some UV radiation. Usually the amount of UV radiation you need is about 20 minutes, 2 to 3 times a week. That's it, not much. And you'll be able to make all the vitamin D that your body needs. But you can see vitamin or UV radiation here in our vitamin D. We're using that energy to break the bond between carbon 9 and 10 in the B ring. So that opens up. And then once that's opened up, you can see we have a carbon here that has a double bond to it and a CH3. Well, that'll just spontaneously change to the double bond being on the outside to make this a CH2. And then you'll have vitamin 2 or D2 or vitamin D3. And what vitamin D does is that it helps to increase the amount of calcium that you absorb through your intestines. And so that's why you often see like dairy products promote now with vitamin D, now with calcium. And the idea there is you drink both of those together, you get more calcium into your system. But yeah, they're all steroids, and this one just happens to have to be active by UV radiation. That's our vitamin D there. And now we're going to move towards another type of building block. So for the last 15 minutes, we've been talking about the building block of steroids, those 4 fused rings. The other building block we have for lipids that aren't structural, so these are all lipids technically, but they're not structural or energy storage. The building block is the isoprene, which is this carbon unit in the top left here, 5 carbons. And we use this 5 carbon structure to build different components. If you've studied electron transport chain yet, you might recognize coenzyme Q, CoQ, also called ubiquinone. This is an electron carrier from complex 1 to complex 3 or complex 2 to complex 3. It does both. If all that's foreign to you, well, feel free to take biochemistry too, and we'll talk about that in depth. But that is all made out of these isoprene units. You can see here in the tail where we have 5 carbons, and this N means it just is going to be repeating for some number of N. And these are all hydrocarbons. You have a couple oxygens there, but for the most part, this is incredibly nonpolar, coenzyme Q, and anything made out of isoprene are all going to be nonpolar. So that means they're going to be able to be soluble in the lipid bilayer then. So coenzyme Q or UBQ, when it goes through the electron transport chain, it actually travels through the inner mitochondria membrane to get between complex 1 and complex 3 or 2 and 3. because it's fully dissolvable with the tails. It can interact with the tails, no problem. If it tries to leave the membrane, then water would not appreciate that. Energetically speaking, it's very unfavorable for that to happen. So that's why it stays in the membrane there. Let's look at some other non-structural lipids made out of these isoprenes. Vitamin A, shown here, also called retinol or retinal, depending on what the X group is. This helps us see. So what happens is that when you have retinol or retinal, the way we see is that when a photon hits our eye, this molecule is going to change. And our bodies can notice that change, and we send an electrical signal to our brain, and that we see as light and as image. It's all mainly due to retinol, which is vitamin A. And a fun little fact about vitamin A is that there's a lot of vitamin A in carrots. And so there is kind of like a myth going around that if you eat carrots, it would improve your sight, which there is no scientific backing for that. If you eat carrots, your sight won't degrade as fast, but there's like no proof that it actually improves your vision. And that whole myth comes from World War II, when England had radar and Germany did not. And England didn't want to let Germany know that they had this technology, and they were able to shoot German bombers out of the sky during dark. And to explain that, they had a mass propaganda campaign saying, the reason why British people can see so well at night is because they eat carrots, right? And so that's kind of how the myth was born, more or less. But as far as we know, intaking vitamin A doesn't really restore your vision. It can help it not degrade, though. Another isoprene vitamin here is vitamin E. This is a class of molecule called antioxidants. And what an antioxidant does is that it absorbs what are called free radicals. When you do oxidative phosphorylation, one of the side products that can happen is that you can have an oxygen molecule with an unpaired electron. And these unpaired electrons can go and ruin proteins and ruin DNA, because that molecule just passes that electron to anything it touches, which will break bonds because it's very high energy. And so to prevent that, we have various antioxidants that will go and more or less absorb that electron. It will be destroyed by the electron, and it will help dispose the electron rather than that free radical electron destroying our biomolecules. Unfortunately, there doesn't seem to be any evidence that if you take like vitamin E or other antioxidants alone, like in a pill form, those don't seem to be that effective at their antioxidant properties. It's more when you get antioxidants from like eating actual vegetables, like with everything else combined in that vegetable, that we start to see the antioxidant properties. And one thing that antioxidants can help prevent again is aging, because it's thought that aging is due to us breathing oxygen and free radicals slowly destroying us from the inside. And that's kind of why we age. So the idea is antioxidants can help prevent that. Here, vitamin E, they are going to help prevent your membranes from being destroyed, because you can see long tail going to go right in the membrane and absorb any free radical oxygens. Vitamin K, also an isoprene Non-structural lipid. This is one of the signals that gets released when you have a cut that will help recruit all the molecules needed to clot your blood. Your book has a very cool chart that goes over this and how vitamin K is part of that, but we're really not going to look at that chart. For these, just know like what is the role of these vitamins and whether they are steroids or isoprene vitamins. So let me just pause right here, take a drink of water, and ask if there's any questions so far. All right, let's move on. So here's a good way for you to review. So for these, try your best not to look back at your notes, look back at the PowerPoint, see if you can actually get this, and then go back and check to see if you got them right. So I'm going to give you like a minute or two to answer these. And these are similar to type of questions you might see on test 3. So see if you can answer these. And I'll be back in just a few to give the answers. Oh, you don't have to actually answer those in chat. Feel free to just like write them down, and then I'll go over the explanation. All right, so let's take a look at these. So number 1, cholesterol is a steroid hormone. And that's actually false. So cholesterol is made from a steroid. That is true, but it's not really a hormone. It has structural components. That's what it does. It's more of a structural molecule in that it's in the plasma membrane and helps to modulate the fluidity of the membrane. A hormone is going to go through your body and start different cascade processes happening. So it is a steroid, but it's not a hormone. What is vitamin A function in the body? Photoreceptor. This is what's in your eye, and when light hits it, it goes through a chemical change, and that's how we see light. What do glucocorticoids do? They have a couple of functions. One function is metabolism. They help to control the rates of metabolism. They also help do inflammation. And then vitamin D, is that a steroid or an isoprene? Vitamin D is a steroid derivative. So these are just some examples of questions you might see on your test 3, which is not next week, but I believe the week after next. I got my dates right. And I'll talk more about that next week though. So going back to structural lipids now, let's actually talk about what's happening in the lipid membrane. So the membrane is fluid in that it's not a solid. If it was a solid, we would all be dead. But it's not a liquid, or else it would just flow away and would all be dead. So it's like has part solid, part liquid properties. And inside your membrane, lipids are free to move. And there's two types of movement. There's transverse movement, where you go from one layer to the other, which I have is very slow. And then lateral movement, where you just stay in the same layer. So why would a transverse or flip-flop movement be incredibly slow compared to a lateral diffusion movement when you're just going in the same membrane? Any ideas about that? Oops. Well, again, so let's take a look at how the lipid is constructed to really understand this. So the lipid in your bilayers are made out of two parts because they're amphipathic. You have a polar part, which is that group. And then you have the nonpolar part, and then the polar part. So to have an actual lipid go from one side to the other, you would have to take this polar head group and make it travel through the nonpolar tails. That is incredibly unfavorable, very energetically unfavorable to do that. And the rate's so slow that in your lipid bilayer, you might have 1 to 2 lipids do that, per day. And you're talking about a place that has like hundreds of thousands of lipids. So it's an incredibly slow process. You do have enzymes that will do this for you, and those can work very fast because they help to shield the head group, the polar head group, when they're going from one side of the layer to another. But without enzymes, this is incredibly slow. While lateral diffusion, the polar head groups are interacting with the water up here and interacting with the polar head groups at the top, so they're fine. And the tails, they're going to be interacting with nonpolar tails the whole time as well, so they're also fine. So that's why transverse is incredibly slow versus lateral, which is extremely fast. Lipids can move really fast. So not only is there movement in the membrane bilayer, but there is two different states you can be. One is like your normal kind of liquid solid state, and the other one's your more solid state. And the switch between these two is transition temperature. Or I guess it would be melting temperature, not TS. And so as a cell becomes colder, as it becomes in more colder environment, one of the things that will happen is that your lipids will go from their normal liquid-like state to form a solid. And this is really bad because the membrane needs to have fluidity in it. Because as you get colder, your membranes will start, or sorry, your lipid tails straighten out, and you have a lot of van der Waals interactions here. So to prevent that, what we'll see as a cell gets colder is that the actual lipids in the bilayer will change, right? So the colder you go, so as temperature drops, you'll have more short-tailed membranes, and you'll have more double bonds. Because lipids that have shorter tails and are unsaturated, they are harder to freeze. They don't have as good as interactions. While on the reverse of this, as temperatures get hotter, to prevent the lipid bilayer from like completely melting, what you'll see happen is that cells will create longer tails that are saturated because these want to stay together more because of van der Waals interaction. So that's kind of a dance you'll see at different temperatures. What by having these lipids at these different tail lengths, you are modulating or changing the melting temperature. You are changing the temperature at which the lipid becomes more solid-like and more lipid-like. And it's very important for the cell to do that. Eukaryotes, as already mentioned today, we can do this via cholesterol. We can just add or remove cholesterol, and that also changes the melting temperature there. So that's some of the considerations we have to remember about the lipid bilayer. It's fluid, it's liquid/solid, but it is fluid, and we need to make sure it stays fluid at all times. Right. So question 3, I just kind of answered. Try and name all the ways you can change transition temperature. So that's by cholesterol and changing tail length. And saturated versus unsaturated. By changing all of these, you can change the transition temperature of the membrane. Now, membranes themselves, certain lipids are only found on one side of the layer. So that's what question 3b is trying to get at. So when a cell is going to die, the way that we know the cell is ready for death is that on the outside layer, so let me draw that. So I'm really exaggerating the lipid bilayer here. So that's the lipid, oops, this part, this whole ring is the lipid bilayer. And when a cell is dying, you have the lipid phosphatidylserine on your outer membrane. And that's a signal to macrophages to come and destroy the cell. And the question is, why do you think PS would work as a signal for death? Or another way to answer that question, or ask that question rather, is why don't cells die all the time by macrophages binding PS on the outside of their cells? So why is this a good signal? And why does this not get out of control? Why aren't our white blood cells killing healthy cells because of this? You want to have ideas about that. Well, the idea here is in normal healthy cells. Normal healthy cells have PS in their membranes, this phosphatidylserine, but it is only found on the inner membrane, right? It's never pointing outside of the cell, because if it was pointing outside the cell, that cell would die. So when the cell is dying, it will start to show PS on the outside, this phosphatidylserine, and that is the signal for your white blood cells, your macrophages, to come and destroy the dead cell, while completely ignoring any healthy cell, because phosphatidylserine is normally found on the inside leaflet, not the outside leaflet. So that's why PS is such a great target for death in a cell, because it's only shown on the outside if something is wrong with the cell. So questions about that idea before we move on here? All right. So let's continue to talk about the membrane, but this time we're talking about integral membrane proteins. And so integral means they have to be part of the membrane. They can never not be a part of the membrane. Here on the bottom left, we're looking at hydropathy indexes for membranes. Hopefully you remember what these are. Basically, a positive number means that you are hydrophobic. A negative number means you're hydrophilic. And we're looking at two different proteins that are both integral membrane proteins. And what we can see from here is that to interact with the membrane, you need a large region that's hydrophobic. So it's the hydrophobic regions that are wedging themselves inside the membrane to interact with the tails of the lipids. But you also need hydrophilic regions to interact with the water and the polar groups. And that's what we're showing here. Here are all of our amino acids. You can notice on the outside, for the most part, you have your hydrophobic amino acids, or sorry, hydrophilic. That would be like your purple. So these are polar. Your red is charged. Your yellow are hydrophobic. So if we go inside the membrane, you can see you only have like hydrophobic with threonine mainly just there to interact with the polar head groups. And then once you leave, you have more charge residues again. So if a protein is integral, what that means is that it's usually going to be a transmembrane protein. That is, it goes all the way through the membrane and it can never be taken out of the membrane. Once it's in the membrane, it's there for good. Here are just some examples of what transmembrane proteins look like. A lot of helices or beta barrels, that's what this is called. It's a barrel made out of beta sheets, so that's a beta barrel. Here are 3 beta barrels coming together to make one protein. You can notice there's like a hole between these beta barrels. That's because these proteins, a lot of the times, they will be used to transport polar molecules across the bilayer. So normally charged or polar molecules can't go through the plasma membrane because they can't travel through the lipid tails. However, these proteins, these transmembrane proteins, the outside of the protein can interact with the lipid tail, while the inside of the protein could be polar, so it will interact with an ion or a small molecule. And we see that a lot in biochemistry when we look at membrane proteins, that they are designed to really bring across one type of ion or one type of molecule. But that here are just some typical representations of transmembrane proteins. And another way that proteins can interact with a membrane is if they are modified by lipids. And these lipids are linked to the protein, and then these lipids themselves insert themselves into the bilayer. And here are just some examples of different modifications that you can make to your protein. So here's our protein down here, and here's our modification. And based on what type of modification you have, it's either called a farnesyl residue. That's when you have 3 isoprene units. That's what these building blocks are, isoprene units. Or a geranyl residue. That's 4 isoprene units. So that's one way that you can attach a lipid to your protein. Lipid goes into the bilayer, and now you have a protein associated with the bilayer. You can also just put straight up fatty acids on your lipid. So here's mystric acid and palmitidic acid. Again, you just attach these usually through the head group. You attach the head group to your protein. You stick the lipid tail in the bilayer, and now you have a protein that is associated with the bilayer because of these lipid modifications. So that's another strategy that's different than integral membranes. And last, another strategy is GPI-linked proteins. What GPI means is that they are attached, and the word is canceled, is not showing for you on the bottom, I'm sure, to phosphatidyl inositol. So phosphatidyl inositol. Oops, that's an L, that's an I, which is shown here in the green. So here we have our protein. We're attaching this to phosphoenolethylamine, which is attached to a bunch of mannose and then glucose. Then it's attached to this phosphatidyl inositol. And here are our lipid tails. So we have here's our glycerol backbone, right? And here the R groups are fatty acid tails. And so these GPI-linked proteins can go and interact with the plasma membrane again, as their tails will just go right in there, and you'll have a big protein just sticking off. And so here's a nice image that's showing the different types of proteins. If it's transmembrane, remember that's what that's called when a protein goes all the way through the bilayer. That's an integral membrane protein. It cannot be removed. You also have something that we really haven't talked about yet. That's your peripheral membrane proteins. These proteins can either be associated with the membrane or not. They come on and off all the time. Those are peripheral. Or your lipid-linked proteins, and this picture is extremely weirdly drawn, but these lipid-linked anchored proteins will have a lipid in the membrane connected to the protein, or it could have this giant GPI linked to it as well. And these, once you're lipid linked, you usually don't come off the membrane either. You're usually stuck there if you're a protein. But unlike the integral membrane proteins, it is possible to cut off the lipid tail So you hydrolyze the lipid that's attaching you to the membrane. So cut. And then the protein is free to leave because nothing is attaching it to the membrane anymore. So that is possible. And we do see that. That's can be used for signaling. Can also be used to do different processes inside the cell. But these are your three main classes of proteins that associate with the lipid bilayer. Right. So any questions up to this point? All right. If not, it's roughly 250. Not enough time to really go into another topic without going over. So I will cut it here. Thank you for coming. Thank you for watching on YouTube. Always appreciated. I will post homework online due next Friday, as always. Next week, we'll continue on our journey of lipids and enzymes and all that fun stuff that you know and love. Well, maybe not know yet, but you will know. And maybe not love now, but one day. And we will continue on from there. All right. So that's all I have for you. So hopefully you have a good weekend, and hopefully I'll see you Monday. Take care.